Good to have you here. So
It's great to be here.
it's good to see you again. You've been so let's see, you were in Baltimore this week. We had a big conference that was in town.
The annual American Society for Cell and Gene Therapy.
All right. So it was a big party.
8 to 10,000 people.
All your closest friends. Well, we appreciate you making it. For everybody, this is our second video together. You and I have done a couple of presentations for some groups, but this will be the second time that we are together. This is, really an update from our first video about a year ago. You were in my office at that time, and now you're in my home. So we're getting closer, as it were. But I wanted to, first and foremost, thank all of our supporters and donors. A lot of what we're going to talk about today is a result of those donations. And, today was really just a kind of a conversation for us to talk about some of the progress that's been made in the last year. And then I think even more excitingly, kind of the direction that we're going in.
Yeah,
So,
great.
all right, should we get started?
Let's do it.
Okay. All right. I think first I'm gonna cheat here and use my notes. But I think first I would love to just, if we could, just because it's been a little while, just maybe a short refresher on what the research is, a little bit about CRISPR and why we're using that for the DMD BMD research that we're doing.
Sure. Okay, so let's put it all in context. 1 in 10 or 1 in 11 people will have a genetic disease.
Yep.
These are caused by single changes or changes in the DNA. That can result in basically 7 to 10,000 different genetic diseases. As a whole is what that number represents.
But I mean the 1 out of 10, that's what that number represents. Yeah. So common, but then you subdivide it, and it's obviously a whole array of different conditions that can exist underneath that, from mild to severe.
Exactly, yeah. And any number of diseases and the severity and things like that, right? There's so many changes. And all it takes is a single change, a single base in the DNA to cause one of those diseases. What if we could correct that? What if we could change that? That's what CRISPR allows us to do. So for the first time, we are able to go in and fix that change or make that change. And that's essentially what the technology allows us to do.
Yep. So specifically for BMD and DMD, when we're talking about CRISPR, DMD is one of the largest areas of the human DNA strand, right? And so, and obviously, DMD and BMD can be absolutely catastrophic diseases. CRISPR will allow us to fundamentally correct to some degree and significantly change the direction of the disease, correct?
Absolutely. So the potential is absolutely enormous. As you stated, the disease is a really devastating disease. Affects about 1 in 3,500 to 1 in 5,000 boys would be DMD. It's a little bit harder to know what those numbers, the precise numbers are for BMD, but much bigger.
Right.
So what we can now do is, and the other thing that you mentioned is absolutely correct too. DMD is a really large gene, one of the largest in the genome. And if you think about that, that actually means that there's a lot of places where something can go wrong.
Right.
So there's a lot of different types of mutations. There's thousands and thousands of different things that can lead to DMD or BMD. But the gene is actually interesting, and it has properties that allow us to correct large fractions of patients with a small number of different products. So even if there's 10,000 different mutations, we don't need 10,000 treatments, one treatment for every mutation. In fact, if we have about 6 to 9 therapeutics, we could capture probably close to 90%
Yes.
of all DMD patients. And then obviously a similar number for BMD.
So as I understand it, the major difference here as well is that CRISPR effectively creates a platform so that basically 99.9, let's call it percent of the research is replicable across multiple mutations or changes in that genome versus small molecule science, which is really looking for a needle in a needle stack, so to speak. So this is a very different approach and why there's so much promise in it.
Totally. So the traditional like aspirin type of small molecule world that we've had for the last 120 or so years is like that, where it is one, we need to find that needle in the haystack, and that needle may not exist in the haystack.
Right.
With genetic medicines like CRISPR, what you're talking about, uh, the fact that it's in our genomes and we know that this problem is caused by this, we know exactly what we need to do to fix it.
Right.
And then the other thing about CRISPR that makes it really this platform, as you were mentioning, is that the vast majority of it doesn't change. And all we have to do is essentially tell that where that problem exists.
Yep.
And that's a small, percentage of the entire thing. And then what you can do is you can imagine, well, if there is a specific mutation, you have that little, small percentage tells it, we design that to go after that mutation. If there's a different mutation, then we just change that slightly, and that makes this platform unbelievably powerful.
Right.
It's based off of the DNA, and we know what causes the problem, and we know how to fix it.
That's great. It's amazing. So let's take it from kind of 10,000 feet to get a little bit closer to ground level here. We over the last year, Hunterian Strongest Hearts have been very focused on a cellular type of data, and I know we don't want to get too far ahead of ourselves on this, but some of that data is going to be things we're talking about in future conversations. We're getting really close to some of that being available for everybody. Let's talk a little bit about kind of the next phase. So we've had a fairly successful phase one of this, and the original plan was phase one was the cellular level, phase two was small mammal, phase three larger mammal / primate, and then the hope is of course to get to a therapeutic clinical trials. We're moving now into that small mammal testing phase. I think this is important. What are the actual goals for small mammal testing, and why is that important? I think anecdotally all of us know that we need to test things before we do them, but maybe you could talk to us a little bit more about just why actually is it so important, and how is that data driving where we're going?
Yeah, absolutely. So I mean, as you mentioned, the goal is to go from an idea really to a therapeutic in a human. So what we do is we start off small, and we basically, get a little bit closer and a little bit closer, a little bit closer. And along the ways, make sure that whatever we're testing works and that it's safe. So as you mentioned, what we've been working on is doing the cell testing. So those are cells that are growing in a dish in a lab, and either human cells or mouse cells or whatever kinds of cells. And we go in and we correct the mutation. So we, as we were talking about before, we can do that.
So real quick, if I could just interrupt. So the cells that you're working on, you've already given some of these mutations to, and then you're actually correcting those cells. That's kind of phase one, is to correct those mutations in a lab cellular environment.
So actually what we're doing is we're doing it in the same place.
Gotcha.
Where the mutation is. That exactly what you mentioned is the phase that's actually going to be coming up for us next.
Got it. Okay, cool.
And we can talk about that later.
Yep.
So the small mammal thing is, as you said, is a standard part of the research path. Typically it's rodents, and out of that, usually it's mice.
Right.
One of the reasons why mice are in particular really good for DMD is there's actually a mouse out, a well-known established DMD mouse.
Oh, it's something that occurs naturally in mice as well as in humans. So got it.
Exactly. It
Yep.
was a mouse that people were studying. They saw, what it was. that it looked like it had the phenotype of DMD.
Yep.
They later found out that, yeah, it actually has a mutation in the DMD gene. And so what we do is we take that mouse was identified like in 1977 and has long been established in the research community. So what we do is we take that mouse and then we correct it. And so like there's a big step up from going from the cells where we're just showing that we can target specifically the right regions of of the gene, to now showing that here's a diseased animal
Yep.
that we can now cure and what the levels are.
Right. Living, breathing creature that actually is going to replicate cells, and
Yes.
and the treatment is going to effectively show, yes, this does or doesn't work the way we expect.
Exactly. And
Yep.
so the two things that we're constantly evaluating, safety and efficacy. Those are the two things, right? How safe is obviously, self-explanatory. The thing is, we don't want something to be toxic. The other thing is we want it to be efficacious, so we want it to work. We want
Right.
it to
Yeah, they're not mutually exclusive effectively.
yeah. It shouldn't be.
Yeah. Unfortunately, sometimes it is, but well, so I think you answered my next question, which was, why are we testing on specific small mammals? And just to kind of to recap, because this is actually naturally occurring in these animals, and so we can take animals. I believe some dogs show a similar phenotype to DMD that's present in humans. So rodents, mice, dogs, being able to correct it at those levels gives a really good indication of, okay, if we can do it here, then the correlation would be that very highly likely we could do it in humans. Is that a fair
Yeah.
summation?
Yeah. No, I mean, I think um, it's there's a lot of information that we can get from these studies.
Yep.
Everything from whether or not the actual strategy is working. The great news for something like DMD is that people have shown that this works.
Right.
And people have done things in mice to show that they can cure from DMD. And as you mentioned, there's other models like the dog. There's several dog models. They're all naturally occurring
Yep.
mutations. And people have shown that a CRISPR treatment can effectively cure either a mouse or a dog. The big problem is now getting that to humans, and for reasons that are technical, people have not been able to do that in a way that is safe
Right.
and effective.
Yep.
And that's the really the technology that we have
Right.
allows us to go from that to something that works in humans.
That's right. And then I think, just because we're talking about it, there's actually kind of a next step that you've talked a little bit about with me, which is that you can actually start to do work genetically with things like pigs and other animals to actually replicate very specific human types of mutations, to even take that testing to the next step.
Absolutely. So like all the mouse and the dogs, as we were talking about, are naturally occurring. They just are born and happen to have a mutation.
Right.
One of the things that CRISPR allows us to do is actually engineer a mutation. So it's kind of interesting. So now we're talking about CRISPR as a tool.
Right.
And what we do is we use, in the same way we're sort of manipulating a genome, what we can do is manipulate the genome of an animal, and we can create a mutation that exists in humans in that animal.
Yep.
And then we can treat that animal and have something that's closer to the human, right? So that's an engineered model. As you mentioned, the mini pig is actually a really good example of that, where there's a model that exists. Multiple groups have created models that are exon 51 deletion, for example.
Yep. That's great. So that's kind of where we're going, right? And these are things that, Strongest Hearts, our next phase of fundraising is targeted specifically towards the small mammal phase 2 component. But obviously, if we're going there, there's been some successes, and you feel like this is a worthwhile thing to do. So maybe we could talk just a little bit about what you're seeing so far and some of the successes or data that you have so far in terms of what's gone on in the last 12 months.
Yeah, so the biggest thing that I think is I think what is the most exciting is that we're not talking about a small subset of either DMD or BMD mutations. So what we've been able to do, and part of the things that we're talking about is we're actually in parallel looking at the vast majority of mutations that affect patients.
Right.
So
When we say we, you mean Hunterian, just to
yes,
be very clear. Yeah, not
yeah.
not the royal we,
No,
specifically
yeah, yeah.
specifically Hunterian.
Specifically Hunterian.
Yep.
It's specifically because of strongest hearts. So one of the things that, like, as you know, when we went through this path, the idea was that we want to address the disease
Correct.
as a whole. We want to, like, the goal is to reach the entire community.
Yep.
Normally, that would not really be possible, to be honest. In the world of small molecules, you would have to go out and try and find whatever number of things. It hasn't happened yet, and it's not even clear that that's possible. The reason why we could have that conversation is because of CRISPR, and that allows us to now look at, hey, this is a platform, this is a way to finally address many types of mutations, and particularly in DMD and BMD, things like deletions and duplications, and things that are much more complicated to address than the types of therapeutics that are on the market, at least right now.
Mhm.
And a big problem with the things that are on the market right now is both certainly lacking efficacy, and things that are certainly questionable safety-wise as well.
Right.
And so, this is really the first, opportunity the technology is allowing to address these types of mutations.
That's awesome. So again, just to kind of summate here, you guys have not been looking at, just as you were saying, like an exon 51 or an exon 3. And one of the goals when we started talking, a few years ago about working together was the idea that, yes, of course, my family is directly affected by one mutation and one exon, but the reality is that there's, thousands of people who are affected across this whole range. The data that you've gotten so far, and I know there's more data coming out in the next 30, 60, 90 days, but what you've been able to see so far is that this methodology does in fact work across that broader range as we had discussed, and the cellular data has supported that original hypothesis.
Yeah, so that's the great thing about CRISPR. In some sense, it's actually easy, because if we know that there's a mutation and we know that, sort of what the fix is, the result that it works is in some sense not surprising.
Right.
The thing that's surprising
It would be
is that we
surprising
can
if it
do
didn't,
it.
right?
Yeah, well, yeah.
Because theoretically, going back to what you said before, if we know where the problem is, we know what the fix is, you're actually just validating data that you already have in that sense.
Absolutely. The thing
Yeah.
that's surprising, and the thing that I don't get over, is when you see it, even on a daily basis, just in action, right? You see how easily it goes, and it modifies the gene. I mean, this was the type of thing that, 10 years ago was difficult, if not, not quite impossible, but the technology was not there, and certainly not there in a way that was scalable.
Right.
And then CRISPR comes in, now all of a sudden, you can do this. And here I am 10 years later, and it's still like the type of thing that is unbelievably exciting to see. And, it all makes sense.
Right. Yeah. And to me, and I've talked about this, so going off on a bit of a tangent for a second, but you and I have talked about this before. This actually is all in line with just kind of the original theory of CRISPR that won a Nobel Prize in
Exactly.
2001 and 2009 and so on and so forth, where, now the technology has caught up to the theoretical, and you're able to validate these things in a lab environment much more effectively.
Yeah. So yeah, exactly. So you're talking about, like, like the ways that we've been looking to edit DNA.
Yep.
That starts really like in the early 70s, work done at Hopkins. That's really the first generation of editing. And, so we've come a long ways from 1970. The technology was developed, some newer technology came out really in the 2000s. And then CRISPR just revolutionizes that thing. That is in 2012,
Yep.
is when that comes, and that's the wave that we're seeing now.
Right.
And it's materialized in ways with some of the most fastest developed approved therapeutics ever.
Yep.
So sickle cell is the big example.
Right. I was going to go a little bit off subject there, but I would actually love for you to talk about that because when Strongest Hearts is out talking to our community and to our donors, we talk about how fast CRISPR is comparatively to small molecule and traditional pharma, right? And it's one thing to say it, but there's actual real world examples now, like sickle cell and some of these other things that are now FDA approved. Maybe just like for a minute, like talk about that because that's the path we're going down.
So let's start with like what the small molecule, because that's the world we're coming from, right?
Mm-hmm.
So on average, it would take 20 years and 2.4-ish billion dollars
With a B.
to get from a bunch of chemicals to something that is an approved drug on the market.
That also won't kill you.
Yeah, like aspirin and things like that.
Yeah.
And that's in the best case scenario if that thing actually exists. There's no reason that there's a chemical out there that we can take that won't kill us. It'll go to the right place. It'll actually have some effect.
Right.
It may not even exist. So we may be, and it's part of the reason why that number is really so high, is because it covers a lot of failures that are rolled into that.
Right.
Right. Like all the opportunity cost of all the programs that didn't work out. So $2.4 billion for something that takes 20 years. So if we think about that 20 years, the first 10 years of that is trying to identify that compound. So going from like millions of things to one, and then the next is basically advancing that compound through cells and animals, large animals, and then into human trials, phase 1, phase 2, and phase 3, then approval. So that's 10 years. The beginning half is 10 years. If we now look at CRISPR, that, what would really have been 10 years is been collapsed to a couple of hours,
Wow.
in a day.
Wow. Because we know where to go. So and we know the methodologies and those sorts of things.
Exactly. So what happened in the early 2000s, we sequenced the human genome and we've been refining that. So we know what essentially the reference is for a human. We know what the normal is. At the same time, people have been cataloging these mutations. So we know things, when this changes, we end up with this disease.
Right.
So we now know what right is and what working is and what broken is. And if we can go from one to the other, that's kind of the final loop in this, really this, type of scientific, it's really multiple scientific um, uh, areas of like study that are all intersecting at the same time.
Right. Like Human Genome Project, CRISPR.
Yeah.
And then adding to that, and this is something that I'm for my other life kind of excited about. Some technologies are also accelerating this, correct? I mean, we've talked about the PCR machine AI. Maybe you could talk a little bit about the technologies that are actually really helping to push this along at an even faster rate than ever before.
Absolutely. So the biggest thing I think everybody is hearing about is going to be the artificial intelligence, right? So there's no question that that is making huge impacts in how we do research, how quickly we can move things forward. So we were talking about, how long it takes to develop a drug. Well, now, we have one thing like, CRISPR that collapses basically decades of work And also the chances of something being successful are way, way, way higher. So we've seen that with sickle cell. We've, with an approval that took basically 5 years instead of 20 years and, $2.4 billion. So the other thing that I think now is really making an impact is, as I mentioned, artificial intelligence. So that's something that we've been using. So particularly with DMD, one of the things that we do is you can imagine there's a lot of places in the genome. So how do you figure out where's the best place to go? So what we do is there's slightly different levels of artificial intelligence. Generally, like artificial intelligence is a broad like category. You then have something called machine learning, and then you have something called deep learning. Deep learning is what we've used on the genome and with the sites to figure out what are those best candidates. And what that allows us to do is really speed up something. So instead of us needing to look through tens of thousands or hundreds of thousands of potential sites, we can now look at something like a dozen or something less. And again, that's something that can now be done in a really short period of time. It's really transformative. All of these things coming together.
Right. And now you can also measure some of these things in a different way as well, right? There's new technology that not only are you able to identify more quickly, but the precision with which you can measure some of these things has changed dramatically too.
Absolutely. So that's another cool thing that we've been developing and really the support over this last year. One of the things that we absolutely wanted to do was have the most precise way of measuring DMD. So we want to know when a patient comes in, what level of DMD do they have? And if we correct it, what level do they get to? That's ultimately going to be like basically the therapeutic window.
Yep.
So the most precise way one wanted to determine this would be to count go into a cell and count the molecules. How many DMD messages are there in that cell? Well, we in fact have a way to do that, and the work that the foundation has supported allowed us to get an instrument that we can now measure that. So what we can do, we can take cells either from experiments. It could be from cells in a dish or cells from a mouse, a large animal model, or patients in the clinic, and measure down to single molecule.
Wow.
How much DMD do they have, and and what the change is.
So this is replacing things like leveraging CRISPR and these types of tools. One of the things as a parent that we've struggled with is, things like biopsies and more traditional methods, which by the way are also highly inaccurate because it's also based on where the sample's coming from, when you're getting it, those kinds of things. So these instruments, just to make sure that, I understand, but that everybody who's listening does as well, what you're able to do is identify the areas more quickly, and then once identified, measure the efficacy of the treatment, and then you'd be able to take somebody and say, you have DMD or BMD, here's how effectively you are by that, and here's what this treatment will do in terms of efficacy for you.
Exactly. And actually, the next phase that we're going into right now makes that even better. So instead of having to start with the patient and get a biopsy, what we can do is we can take a stem cell and we can now engineer. We were talking about earlier about engineering, like a pig to have a mutation. Well, what we can do is we can take a patient can come in, they can get their mouth swabbed, we can figure out what the sequence of their gene is and what mutation they have. Then we can engineer that into stem cells. Stem cells have the property where you can turn them into other cells. So what we're now starting to do is gear up to do muscle stem cell modeling. So we engineer the mutation into the stem cell, and then we turn that stem cell into cardiomyocytes or skeletal muscle in a dish. And that muscle carries that mutation that we've engineered. So now we tie that together with that instrument, right? We can now measure precisely the molecules of DMD from that mutation leads to. And when we correct it, how much does that improve? What is the level of improvement?
Well, so you're doing it at an individual basis effectively.
This is really personalized medicine, like coming together
Mhm.
as a whole, right? People come in, they have whatever mutation, we treat that mutation, right? Instead of thinking about treating DMD or BMD, it's really treat the mutation, right? It's treat the specific problem.
Yep.
And so that's the plan. The plan is to do that, across the board for really any patient.
Yep. So that kind of raises the question then, I mean, this is all phenomenal news, right? And, I think part of the reason that as we were looking for where to put the resources and energy of my family, Strongest Hearts, this was a no-brainer, right? But obviously nothing is perfect. So what are some of the concerns, challenges, as, Hunterian's been around for, almost a decade, 8 years, but in the last couple of years, what are the challenges you're concerned about as we continue to move forward here?
So the greatest strength of CRISPR is this ability that we're talking about to go after all of these different mutations and to do it in parallel.
Right.
And as we discussed, that was a really important thing, that we were going to help as many patients as possible. We're not going to just go after the important things. The issue with that, or the complexity with that, is that that's a lot, right? It's a lot of work. It
Right.
requires, resources and managing that sort of, it's a larger team, but overall it's much faster. There's a lot of reasons why doing it in parallel is actually, much, much, much quicker and much, overall much less resources than if one were to do it sequentially and then go through and try and address. And not to mention the amount of time that some patients, just based off of what their mutation is, might not have an available treatment.
Right.
So the complexity is moving from that small molecule world mentality,
Yep.
of one thing at a time, to something where, and we're not in and the reason why DMD is so great is because as a therapeutic target is because the amount of mutations and the percentage of patients that we can address with a small number of therapeutics, it allows us really to make the biggest impact.
Yep.
As quickly as possible.
Right, which was something we talked about early on. I mean, obviously, when we found out about Jack and his mutation, the first thing that you want to do is treat that, right? Because that's the natural instinct of a parent to say, we have to fix this. But, one of the things that we talked early on about, and that I was convinced then and now of, is that it's by focusing on the entire uh, genome, by focusing on the, from all the mutations and all the hot spots, which would cover about 90% of the kids, it's going to be a rising tide lifts all ships types of scenario, versus a lot of where the focus has been today, which is on, exon 51,
Yeah.
which, yes, it's the biggest hotspot, but it's also 16% of, 13% of, making me look bad. But 13%, right? So it's the biggest hotspot, but it's still, just above single digits in terms of the group that would be treated with that. So the complexity is that you're going after more.
Yeah.
But the benefit then of that challenge is that the more it will create economies of scale, for lack of a better term, and actually treat a much larger group of people when it's done.
Yeah, that's exactly right. So what the dream is, and the dream not being that far away, is that the moment something is noticed that is wrong, a patient is sequenced, right? We figure out what that mutation is. So what we need to do is we need to cut the time, the sort of diagnostic odyssey
Yeah.
thing needs to shrink,
Yeah.
right? It really should be, down to zero.
Right.
And then you can imagine, once you know what mutation a patient has, if you have, let's say it's half a dozen programs that can cover all of those, that patient then just slots into
Right.
one of those
Right.
programs. And what we would do is once we sequence, we know exactly what their mutation is. We would then engineer that into the stem cells. So that allows us to now validate that that mutation, this product is going to correct it. This is the level of correction that we anticipate. And then the person can actually get that treatment. And that way, like, it's all going on in parallel. The next person that happens to come and have a mutation, maybe it's not, maybe it's something different, maybe it's the same one. It doesn't matter because we're basically, we've created a net where we can capture the most numbers of patients.
Yeah. Yeah. No, and I think the dream, again, I've talked about this in other forums and I've talked about it on our blog and things like that was, our journey was nearly 6 years long, right? And I think that's probably a little bit shorter for
That's the
children.
average
I know, right? But I
for
think
rare disease. Yeah.
for rare disease in general, and I think it's a little bit shorter for DMD because it's more symptomatic earlier. But what we're really talking about is a kid has a swab when they're born or potentially even one day in utero, right?
Yeah.
And they're receiving that treatment almost immediately. And, for some kids, then it becomes basically something that's a non-factor in life. I mean, that's really nirvana, right?
Yeah, and you've hit on it, right? Like the quicker you can detect something, the quicker you can treat something.
Right.
So everything exists right now to do the detection, right? It's really just will and implementation.
Right.
That's something that people can do. It's not what we do, but that's something that, and it doesn't, that's going to require effort and getting the community, like, to enforce that, get that in play. The thing that's been missing is the therapeutic, right? Having like
Right.
now, if you get the diagnosis, what are you going to do?
Right.
That's now there, right? That final piece is now available, and it's available across the whole, spectrum of mutations.
Right.
And it would be a shame in this time when all of these things are coming together not to help the maximum number of patients.
Yeah, that makes sense. Well, I'm gonna wrap up on this one. You know, you obviously had a ton of success so far. I think we've been, speaking from a strongest heart standpoint, tears of joy after a couple conversations, which I know makes you uncomfortable, but that's part of this process too. But I think what I would really love to talk about, and I think you were touching on it, is bringing it kind of full circle. Where do you envision really, 10 years from now, we're sitting here, we're having a conversation. Where have we gone over 10 years? Where do you envision CRISPR taking DMD, BMD, or really any rare disease, right? But, for the purposes of what we're doing today, where do you see this really going? What would your vision be if you could, do it without having to worry about anything else?
Yeah, so I mean, I think you hit it on the head. Of course, DMD and BMD is where we are focused initially. Um, that is likely going to provide the path for other diseases. So it's important to like know that it's not just we're trying to cure DMD. We're trying to address genetic diseases as a whole, and DMD is a great way to do that, to get to other diseases. The ways that we're addressing DMD is a lot like the way you would address other diseases.
Right.
So the ultimate goal, right, is somebody's born, and if there's something wrong, we find out what that is as soon as possible. The next thing is having a treatment that is available to address that. Is there a way to correct that? Clearly things in like DMD and BMD, you want to treat early. You want to treat before sufficient damage has kind of gone past the point of no return.
Right.
So clearly earlier is going to be better. Dosing is likely going to be lower, and safety is likely going to be better. There's all kinds of reasons why treating earlier would be preferable. And then, that person now, which, the dream would be if somebody that was, constrained to live in a wheelchair grows up and has a normal life.
Right. Right. And they're aware of their condition. So, later in life when they decide to have children, start a family. We're also able to potentially make different choices with genetic counseling in terms of how families move forward as well, which, for all intents and purposes could actually stop a disease from being passed on or carried on as well.
Absolutely. And the amazing thing is everything that we have talked about already exists.
Yeah. Yeah.
We just have to get there.
We're going to do it. So,
Yeah.
well, I keep looking at my phone. I promise I'm not checking my email. But, I think one of the biggest things that we appreciate and that we, the Strongest Hearts in Hunterian, had committed to with all the donors was these conversations, really making sure that we're sharing with everybody because I think it's important, right? And, this has been hopeless for a lot of people for a long time. And I think what we're really seeing, especially with some of the other therapeutics that are coming out that deal, non-steroidal things that are dealing with some of the symptoms today, is that there could now be a real bridge between this side of the river where we are, where, treatment options are limited and where we're going.
Absolutely.
Yeah.
Absolutely.
Well, listen, Dr. Vinny, I appreciate it. You are at the end of a 7-day conference. So the fact that you showed up to do this, we can't thank you enough. For all of our supporters, everything that we talked about today is a result of the dollars that you have donated, the effort that you have put in for our volunteers, and for everything that you've done for us. To continue to stay involved as we move forward in our research. Please click the link, donate, volunteer time, spread the word. But we thank you so much. And we are going to be doing one of these about once a quarter now, I think was the goal.
Sounds great.
Yeah. And as we get more data, that will be shared. So again, we thank you all so much. And Dr. Vinny, thank you.
pleasure.
It's always good to see you. And I know you need to get home to your wife and your Frenchie, right?